Well, welcome to a podcast where we are sat with some fine human beings. Why don't you just start with like a very quick of this is my name, this is what I work on, and this is why I work on it.
Hi, I'm Darshit Patel, and founder of a company called Decode Age, and another one that's coming up, still in stealth, goes by the name of Regiome Therapeutics. I work in the area of longevity consumer and longevity diagnostics and therapeutics, actually a lot of it, but hopefully you'll all check out and know what I'm trying to do. Uh, I work on aging mainly because of two reasons. One is more subjective and one is more objective. Subjective because I think that there is a billions of probability worked out to make sure that I'm living on this planet and I don't want to go. So I want to stay here, so stay as long as possible. Objective because I feel that humans are innately meant to solve puzzles by looking at patterns, and aging turns out to be one of the biological puzzles that has not been solved yet. So I'm here to solve that. So yeah, that's the what's and whys of what I'm doing.
Hello, so this is Jagan. I'm from Bangalore. I work in Decode Age as a bioinformatician. So I'm currently studying and understanding gut microbiome and its role in aging. My focus is more towards metagenomics, more focused on pipelines and algorithms for biological data. So I'm currently, working on understanding these algorithms, pipelines, and like more focused towards scientific tools. How do we make them reproducible, robust, and yeah, I'm more focused towards the technicalities uh, uh, of uh, of the biological data.
Hi, my name is Suramya. I work at the Indian Institute of Science, where I'm setting up the Longevity India initiative. And I primarily work on the cell biology of aging, where I look at how and why aging mechanisms change in these cells. And now we're slowly taking that learnings and information up to animal models and humans. We're also running a population study to understand how aging happens in the Indian population and to find out specifically reference values for the Indians. I work on aging because I think there's very little that we know about it. There are multiple pathways and mechanisms that still need to be decoded, and finding and solving all of those problems will then ultimately lead to impact in the wider population, where we can lead to cure of multiple diseases that come with aging.
Okay, well, my name is Yash, and I am one of the co-initiators of ZuGrama. And we're doing this podcast because we are doing an experiment with gut microbiome. I'm not sure what sort of audience will be looking at this, so I have to assume that the audience might not know science at all. So I'm going to ask you a bunch of dumb questions while we are doing this, so you can answer them as well. Why don't we just start with what the experiment is about? Maybe hypothesis, data collection. What are you looking for? Just in a small gist. We'll go into details later.
Before we really get down to understanding the experiment itself, right? I'll just like to give a little bit of background on what microbiome actually is and why people are looking into that uh, globally, right? So we always believed that we are an organism. Well, apparently we are not. We are a meta-organism, which means that there are community of species that are living inside us. Be it your mouth, be it your small intestine, be it your large intestine. Some people are even claiming blood and tissue samples. But every site, there is a certain resident of microorganisms, bacteria, viruses, fungi, archaea, and parasites. And it's not like they have recently entered our body. They have co-evolved with us, and it's a beautiful story how the whole community has evolved as human beings have evolved as well. And wonderfully, it's not just humans. In fact, pretty much all mammals and complex organisms that you see in the world have an innate microbiome. Well, that being said, studying microbiome becomes important, especially the gut microbiome, is because essentially, if you see, your body is divided in two halves, right? And I'm not talking about symmetric or asymmetric, but there's a tube which opens in your mouth and ends at your anus, and that's literally a hollow thing that's between your body, right? It's not closed at either ends. So the maximum interaction of environment is happening through this hollow tube. And whatever is going in, let's say food, let's say air or water or anything that you're drinking, eating that goes into your gut interacts with your microbiome, and your microbiome either digests it, changes it, transforms it, and makes little molecules that is going to be available to your body. So that's essentially the science of microbiome, right? And whatever you're going to eat is going to get transformed by them to make it available for you. And recently, the technologies to study microbiome in much more depth have started coming in. And we are just one of the groups working in the world trying to understand different mechanisms of this microbiome. To sum it up, generally in this world, dietary interventions, probiotics are one of the most tested microbiome interventions. The problem though has been that microbiome is very complex. There are more than 1,000, 1,500 species at a time. So you cannot pinpoint exactly what is causing what change into the biome. And that's mostly because whenever you are doing a study, every person is living in a different house, in a different environment, drinking different kinds of water, different kinds of food, right? Which is why the microbiome intervention studies have not given strong signals so far. When I heard about ZuGrama, and since I was participating in it, I really thought that we could do a very different kind of study. Something that is as much as a citizen science project as it is a science project, right? So the idea is that in this place, which is ZuGrama, people are going to live in a single place for 6 to 8 weeks, right? Uh, they're going to eat similar kind of food, cooked in the same kitchen, breathing the same air because they're all living in the same city, living pretty much a similar schedule of life that they have been. So the exposome is quite limited compared to all the other studies, which means that lots of factors are in control. So the study that we are doing here is to understand what is the short-term effect of dietary interventions on microbiome of people living in a close-knit community, which in itself is a very unique kind of experiment that is going to happen in the world. What we are trying to look at it is, do we really have a strong signal when people are following a similar lifestyle, dietary interventions, and air and exposome that we just discussed upon? Interestingly, for what people have told as microbiome friendly foods is the kind of foods we have asked the kitchen to prepare as well for breakfast, lunch, and dinner. So now we'll be able to really see that what we know about microbiome friendly foods and what we know about the microorganisms and the human mechanisms, would we be able to prove it to a certain extent? Would we be able to get some kind of signal for the dietary interventions, and that too short term and not ultra long term? So that's pretty much, I think, the whole background and why we are doing this.
Okay, so before we get into the details, I just wanted to quickly get what is the hypothesis here?
To check, given a curated food, right, and also given a certain lifestyle habits and giving a controlled healthy dietary habits for a short term, what kind of microbiome changes do we see? Because this has not been studied before. Why? Because we can't create certain environments, right, where people could be in the same space, and also we don't even and we couldn't. When I say same space, there will be a lot of variables, right? So we will not have enough people to get any statistically significant output out of it. So the core thing that we are trying to find is what kind of changes do we see from from this short term from this diet. Like what kind of different changes we see, and why do certain changes occur? Is it happening for there are people who've been taking part for a week and 2 weeks. So what is the difference between people, right? What kind of different microbiome changes do we see between these two groups, right? So, and also it's not just diet, and also people are following certain lifestyle habits like yoga, exercise, swimming. They're trying to do other sort of things. We want to see how all of these are interconnected. Can we find any correlation out of this?
Right, so I think the hypothesis statement then is very simple in a way that given a controlled exposome and diet, microbiome shifts to on the positive side, which actually means that the symptoms reduces for people when they're following a certain dietary guidelines in a certain exposure.
So Ramya, let's take a step back, right? And for the not so science audience, the basic thing that we have here is, okay, there's a bunch of things in my digestive tract which we want to study and look at. So can you just explain what is the digestive tract? Because some people might be confused about what components actually go into it. And we have bacteria, viruses, fungi. Can you make a differentiation between those three so it's clear for everybody who's listening?
Should have revised my biology for this. Okay, so essentially, the digestive tract starts in your mouth where you eat food, and then there is essentially enzymes in your saliva which will break down the food as you chew them. Then you will take this into your stomach. The stomach is where most of the acids are released to break down the food into smaller bits and pieces. And here it will get broken down in a manner where once it moves to the intestines, which follows the stomach, and from where the entire absorption into the bloodstream will happen. The food is in a state where it can be easily interacted with upon by these gut microbiome microbes which are present in your intestines. So once your gut has it's first of all it's wrapped around in your entire abdomen, and then it also has very specialized cells that are responsible for absorption of nutrients. These could be different kinds of nutrients, fat molecules, small molecules, sugars, and different things. So there is a very specialized mechanisms for absorption from the food for these particular components. And then whatever all the components that are to be absorbed are taken up by the intestines. Whatever is left moves down the digestive tract and is excreted from the body. Now, in this entire process, there are certain enzymes which are present in the human body, and the human body will be able to break down certain elements associated with the food that we take in. However, there will be certain enzymes which are not produced by the native human body, and it will be acted upon only by the microbes which are present in the gut. And Darshit can touch upon more about the different types of foods which are broken down by these gut microbes. And so it's essentially a symbiotic relationship where when we evolved, we could eat different types of food, but because there are certain enzymes that our body does not produce, we then acquired certain bacteria that could help us digest these kind of foods and also absorb the minerals and vitamins and different components that were essential for our body, but our body was not capable of absorbing them or breaking them down. For example, many of vitamins are broken down by the gut bacteria and then they are absorbed into our bodies. If the gut bacteria is not present, we'll lead into a cycle of deficiencies for these particular vitamins. So I'll hand it over to him to explain about what kind of foods are sources for gut microbes to break down.
Yeah, before we go on to foods, so these three types of main things are present, right? Bacteria, viruses, fungi. Can you tell us a bit about what are those three individual roles that they play? How do they live together? How are they interconnected? And we've been using the word microbiome, which essentially means all of these microorganisms in one place. Why do we want to study them together and not separately?
This actually the last one is an excellent question. I've never seen that coming my way, right? It's a very well thought out question, but let's start at the very basic, right? So bacteria, viruses, and fungi, and archaea. These are four different kinds of organisms. It's just like dogs, cats, humans, right? So these are four different kind of organisms. So innately they are meant to do different functions. They look different. The mechanism or their machinery inside the cell is quite different as well. Now, one of the largest residents of the gut are bacteria. So let's try to understand what bacteria are, right? Usually unicellular, usually all the time unicellular organisms, right? So unicellular organisms that uh, is known to perform specific functions and known to thrive in specific environments, which means that usually most of the bacteria that are living inside our gut is anaerobic in nature, which means that as soon as you expose them to oxygen, they would not be really surviving outside of that, right? So each bacteria, of course, has its own genome, or what we call the bacterial genome. And each genome will have different genes. That means each bacteria can perform a certain kind of function, right? So this is a basic understanding of bacteria. More of a structural difference, which might help us in later conversation, is that there are two types of bacteria. There's a gram negative and there is a gram positive. Now, gram is actually a stain which is used in lab, but essentially functionally, if I have to describe, gram negative bacteria produces something we call lipopolysaccharide, LPS, right? And gram positive don't. Now, LPS is an interesting molecule in itself. Usually, if it enters your bloodstream, it's going to go to your heart and cause some of the issues there. So LPS is actually one of those inflammatory molecules or that is produced by the gram-negative bacteria, which is why most of the times when you hear the word pathogens or guys that cause infection are gram-negative in nature. But that doesn't mean that all gram-positive bacteria are also good for you. It's just that structurally they are a bit different. So this is what we know about bacteria so far. Then there are fungi, right? We use yeast in our food, right? Usually when there is fermentation happening, along with bacteria, there are fungal communities also being used, be it the production of wine, be it the production of beer, or be it the cooking of dosas, right? Which we have in most of the parts of India. Fungi are colony forming organisms by nature, and most of them are just symbiotic in nature. They live inside you. I think I'll suggest Paul Stamets and his work on fungi to everyone who is interested in this microbial world, right? He explains it so fantastically, I'll never be able to do justice to that. But essentially in gut, fungi plays a certain roles as well, right? Usually you will see they are mold producing in nature, which means the molds that we see around us. They form some of that in our gut as well, but mostly in symbiotic in nature. But some of them are harmful. For example, there's a Candida albicans, which is usually a part of the vaginal microbiome and sometimes the gut microbiome as well. And it's often called opportunistic infection. What I mean by opportunistic is usually they stay in your gut, no problem, completely fine. As soon as you have your immune system going up and down, it will act and it will aggravate the symptoms. So Candida albicans is actually one of the most severe hospital acquired infection, if I have to tell, in terms of fungal infections. Then we have viruses. Thanks to COVID, virus has suddenly become a very famous word globally, right? Before that, no one used to really understand viruses except that when they get cold or sneezing, they would think that I have a viral infection. But viruses are of multiple types. Lots of them live in our gut and they do not infect us directly. But they do this very important mechanism. They infect bacteria and not always in the good ways, not always in the bad ways, but sometimes in good ways also. A mechanism which we know about viruses living in our gut, which I find personally very interesting, is that they go into a bacteria. Sometimes they pick up a gene from a bacteria and go to another bacteria and insert it there. Something we called horizontal gene transfer most of the times. Right?
So, are bacteria also capable of doing that just by on their own? Aren't
So
aren't bacteria also capable of doing
they
the
they are
HGT on their own?
as far as I know, they are capable of doing that. I mean,
So in the gut specifically, it happens via viruses.
yeah, so bacteriophages are quite often known to do that
Interesting.
most frequently. Very interesting paper. If we have time, I'll just go on and tell you a little bit about that paper, right?
But take a step back. You just use the word bacteriophage, so explain that first.
Yeah. So bacteriophages are basically viruses that would infect or target bacteria, right? One important feature we need to realize about viruses are they are not alive innately. They need to enter another organism to activate themselves. And that's what a virus is. So if a virus is going to a bacteria to get activated, then it becomes a bacteriophage. Basically, it's a kind of virus, right? Okay, so coming back to that paper. So very interestingly, we are suddenly seeing a lot of influx of red algae products, right? Going throughout the world. We see people selling red algae as fibers or red algal products in US and XYZ places, right? This phenomenon actually comes from Japanese culture because they have a lot of red algae, and thanks to, I think, Hugo Walter, right? Thanks to Hugo Walter, he has been doing a lot of work on Japanese population and talking about why they live long lives. So Japanese in their ancient culture and still have culinary involvement of red algae. Over generations of exposure to red algae, there used to exist a gene in red algae which is used to produce energy in the plant, which is called porphyrinase gene. This porphyrinase gene was picked up by a bacteriophage from the red algae and transferred to the gut microbiome of Japanese people. And this is essentially the reason that Japanese people are able to extract energy out of red algae. Most of the American population or Indian population or elsewhere in the world do not have that gene in their microbe. So we taking red algae does not make as much sense as Japanese population taking it. So this is one of the mechanisms how bacteria gets involved into the microbiome.
Um, that's a very interesting mention for the specific example of Japanese population and the gene present in their guts, which enables them to metabolize a certain kind of food, which means that whatever information that we can learn from gut microbiomes will be very specific and local to regions, and that will only define what kind of diets will be suitable to that particular population. So in that sense, any food that is being recommended by people globally or across the world may not directly be applicable to you. Is that correct?
I would definitely put stakes on that, right? Especially when things like Mediterranean diet getting too famous across the world. I would just like to take a step back, understand what a local microbiome looks like, and then go more personalized suggestions for the food, right? And this is one concept I think that's coming up more and more after the microbiome studies have started coming up. We do understand that not all populations look similar. And if you really have to design food which is healthy, it should be more area and region specific than a general guideline that is provided to the world.
Interesting.
And
You have a
in
question?
fact, a lot of Indian work that we are doing, we see that most of Indians have very low abundance of certain bacterial family called bacteroides, which actually helps you break down protein. So if you have been generationally vegetarian and suddenly start eating 500 grams or 1 kilo of Cajun in the name of protein, you're doing more harm to yourself than good.
Cool. That was a good lesson for everybody, I guess. But we still need to get to the last question I asked. Why not study them separately?
Very interesting. So, first of all, we need to understand the words microbiome and microbiota, right? Microbiota is the collection of species living in an environment, and microbiome is collection of all the genes across all the species inside the environment. When I'm studying microbiome, I'm looking at what is the potential that or potential for function. When I'm looking at microbiota, it's just who is living in there. Uh, if I have to clarify it more, right? when I say that a country has 100 million people, right? That means that I'm doing microbiota testing, right? Who all are these people, right? It could be Yash, it could be Darshit, it could be Jagan or Suramya, right? But when I say that the country is very good at manufacturing an assembly line, that means I'm going down to the function of the population. That's the difference between microbiota and microbiome. We cannot study them individually. So until 1988, we used to study microbiology with what we used to call culture-based techniques. Culture-based means you take a bacteria, you put them in a certain nutrient environment in your lab and let it grow, right? And that will tell you which bacteria it is. Because you cannot take one cell and identify it. Never, it doesn't happen. What we realized is that only 1% of the total bacteria in the world can be cultured in a lab. What about the other 99%? Thanks to Joe Handelsman and
Hey, sorry, how did people realize that only 1% can be cultured on a
thanks to
plate?
sequencing techniques. I think the first time an environmental sample was taken and studied under the phenomenons of genetic testing, and suddenly we realized, oh God, there's so much bacteria. We never knew what are all these sequences that are coming. It's not even human. It's not even organismal. That's when we realized that okay, there is so much genetic material out there, and not all of them is captured in culture based techniques. That's where the whole metagenomics came about. And now we study bacteria culture independently. That means we don't grow one bacteria and see what they're doing. We just get all the genetic material out of an environment and study where what different bacteria they are. We have done this in water, we have done this in marine, we have done this in Mariana Trench, we have done this on Moon and Space International Space Station, we have done this on soil, human guts, human brains, mice, and all the possible organisms that we know of today. So I would say in the last 36 years, the field has progressed a lot.
I feel like we didn't touch upon why exactly do we need to study the interaction between all of these species together. So if you look at the interactions between them, like Darshit explained using one example, that viruses will infect bacteria. They're capable of modulating them in certain manner. One example of them was that they picked up a gene from one bacteria, put it into another bacteria, right? This could happen like bacteria could pick up the gene from the environment and then transfer it to humans, or they could pick it up in the gut and then transfer it into the gut. And then bacteria would then be capable of metabolizing another food product or not. Bacteria themselves can also cause, and both viruses or even fungus can also interact with the human cells, where they are capable of invading the host, depending on the conditions of the immune system, like he mentioned. So a right balance of the colonies which are there in your gut. Let's assume there are 100 colonies in the gut currently. There will be a colony which is dominant, and there will be one which is being repressed by this other colony, depending on what the conditions of the entire microenvironment is. Now, if the gut is overall very healthy, then there will be a balance in all of these colonies, because every species produces certain compounds for its own resistance and resilience as well. For example, we all know that bacteria produce certain compounds that will inhibit the growth of other species in and around it. That is to its own advantage, where it will promote the particular bacteria's growth, but it will inhibit the other colony from invading its territory, right? So this is how the harmony is maintained inside the gut, and this is how the balance between different species is also maintained. Now, if the environment changes in any form, which means, let's say there is high inflammation, which means the immune system is dysfunctional now and thereby causing a change in this environment, this entire balance can be tipped off. So that can mean that whatever, if one species which was producing a compound that was inhibiting a pathogenic bacteria, that now slowly starts reducing, that means the pathogen can now grow. So the interactions between all of these become very important, and the interaction with the host also becomes very important to understand what the overall health outcomes will be in a person. And these are done using the techniques that he mentioned, where You study all of the genes that are present, what kind of functions they do, what kind of foods they metabolize. And you can also correlate this with the kind of metabolites that are present in the blood of the host to see that how the entire microenvironment changes is affecting the overall outcomes in the host as well.
Okay, so looks like in this study we are looking at managing a very simple way to put it is to maintain optimal colonies of this microbiome. So foods affect those colonies. If in case there is a misbalance in your diet, one could, like if you had too much fungal presence, it could actually harm the microbiome. So help me understand something. So Jagan, earlier you mentioned the hypothesis is that, okay, we need to have a short-term look at certain diets, right? And Darshit said something like, oh, you know what, if Mediterranean food is now popular, it might not be suitable for us, right? So in this study, we're looking at something like, everybody present, the residents of ZuGrama will try to eat a similar set of diet and will possibly see some effects on their gut microbiome. Since we already have that assumption, if, say, for example, I go to a geographic region where the diet is entirely different, won't I just get used to it? My gut microbiome will just get used to the food there, and like everything is now optimal to digest that food thoroughly, and I'm still healthy.
Right. I mean, see, your microbiome is pretty dynamic, right? That means it's subjected to change when you expose it to different kind of things, right? There is certain kind of a static factor that stays throughout your lifetime, but most of it is dynamic. So when I'm shifting out, let's say if I'm going to a different country or a different geographic region, forget about the country. If I'm going to a different city also, right? We all might observe that when we are traveling there's an upset for a couple of days, right? And that's usually the time period. Some take couple of days, some take week, right? But that's the time period when your microbiome is getting adjusted to that particular food, to that particular area, to the particular air and water that you're kind of drinking and breathing, right? So the mechanism is there. Yes, your microbiome is dynamic. It will take time to change, and then it will start settling in, and then you'll be more used to the food that you are eating there. Usually the thing that works best for me is that if you fast for the first 24 hours in a new place, it kind of speeds up the cycle for resetting of the microbiome. I do not exactly understand the mechanism behind it, but that is something I have seen working anecdotally at least.
Okay, when we have tried to craft the diet here, you mentioned earlier we've asked the kitchen to curate a bunch of things, right? So we are making an assumption that there is a set of diet which is more healthier, in air quotes. Um, so how does this work? Like, is it because if I am an Indian, I'll have to only eat certain set of food which might be helpful for me? Because as you pointed out, if my gut microbiome were to sort of get adjusted to whatever new diet it is, what makes this diet healthier?
Sorry, before you take that question, I have another one. Does Where all does gut microbiome come from apart from the ones that have been living in your gut? Is it there in your food, the vegetables, the salad, the water? And depending on the geographical location, let's say assuming that I move to Germany and I start drinking the water of Germany, will then there be certain species that I introduce to my gut that will help me metabolize the food that is there, or are we making that assumption?
100% there is an influence. I would say that because before I started working on gut microbiome, I spent about 10 months or I think almost a year on a project where we were looking at freshwater microbiome of the city of Pune, right? Where we sequenced a lot of water samples of dams, rivers, lakes in and around the city from where the water usually enters the general population consumption. And we saw that there were very specific signatures of certain microbes that were present in this water. And we even, in fact, took some of them from the pipes that were delivering it to the respective households. So microbiome does enter your system through water. That is 100% sure. In fact, there have been papers where they have seen that your social group affects the microbiome profile as well, which means that if Yash, Suramya, Jagan, and I are living in a house for at least 1 year together, we all will influence each other's microbiome through our skin, mouth, gut, everything, right? Food has its own microbiome. There have been multiple papers where certain food items have is known to harbor a microbiome community of their own, and they also enter and influence our own gut microbiome. But that is all at a matured stage of our lifetime. The first exposure for a human towards its microbiome comes when you are getting delivered by your mother, which means that the first day of your life in this world. If you are getting, if you are coming out through a vaginal delivery method, then you would be more influenced by mother's vaginal microbiome. Very interesting, because mother's vaginal microbiome goes through a certain changes just 10 to 15 days before the delivery of the child as well, and that's to make it more what do you say, conducive to the child's microbiome. So it's a very interesting paper that came out, and I was pretty impressed that that happens and how biology plays out, right? But then the first 3 years, the first year is mostly the breastfeeding and the kind of microbes that gets influenced. Most of the microbes that you will find in the vaginal microbiome 15 days before the delivery are the ones that helps the child break down mother's milk into nutrition. So very interesting dynamics plays out there. And as your feeding habits keep changing, you move on to solid foods. Till the year 3 and 4, it changes pretty dynamically, and then it starts stabilizing over the next 5 to 7 years. And then once you hit your near teens, and then it's almost stable until your adulthood. And then whatever changes happens through your environment. The ones that we discussed, the people you're living with, the food that you're eating, the water that you're drinking. But yeah, coming to the question of how can we say that certain foods are microbiome friendly, right? And that's where we really go to a level where we understand food at a very molecular level. We don't see apple as an apple. We see apple as a collection of acids, collection of different substrates, right, which the apple has. And like I said, we talked about the gram negative and gram positive. I told that it might come into influence as we come out, right? Each kind of bacteria can feed on a certain kind of substrate to grow, right? So some bacteria might feed on what we call complex fibers, right? We eat so many cruciferous vegetables. We eat the kales and the lettuces and the broccolis, right? Most of that we don't digest ourselves. That we eat it so that our microbes can feed on them and grow. So these are substrates: complex starch, protein, certain amino acids. Tryptophan, for example, is metabolized by some of the bacteria in our gut to produce dopamine and serotonin. Now these are important molecules for your own body. These will travel through the vagus nerve to the brain and give you some kind of a mood effect, which is why we say that the gut and brain are connected, the gut and mood are connected. When we eat bad food, we don't feel so good. When we eat certain kind of food, we feel sleepy. Why does that happen? It's because of these. So we know a lot of mechanisms where certain foods and their substrates like amino acids, complex starches, and certain kind of carbohydrates, some bacteria can feed on them. This is fundamental knowledge. They feed on them and produce something else. This is how we decide why a certain food is healthy for your microbiome and why it is not.
So looks like we want to change our microbiome optimality in some way through eating some certain amount of foods. This was a good like microbiome 101. Let's go into the study now, right? What exactly is the study? What are people doing? What is the baseline, timelines, everything?
Jagan.
Should I start with aim or should I start with like a study of
Anything.
Okay, so we before the start of study, I think we have people from different places coming here, right? So before the start of study, we want to take their baseline. So baseline gives you understanding about what their gut microbiome was, right? Then for next 3 weeks, they'll be following a diet, which is curated diet. Then during this 3 weeks, I think we have a different set of groups within them. Like there are people who would like to fast, and there are people who are following within the curated menu. They want to follow a certain diet, right? And also there are people who will be who will be following other habits like swimming, gym, yoga. So then end of 3 weeks, they'll be giving us a second sample. So during throughout the week, we'll be collecting a metadata. We built a small tracker app where they will be giving us what diet they're having, breakfast, lunch, and dinner, right? And also we'll be tracking about what kind of physical activities did they do throughout the day. Also, we take a Bristol stool check. If like if they're passing a stool, we want to know what is the consistency of stool, because this has not been understood enough before. Why does a consistency of stool changes? Like I think there is not enough studies on that. And next, also we'll be tracking mood, like how did they feel in the morning before before breakfast, after breakfast, and throughout the day, how do they feel mentally, right? So these questions will give us overview of diet, physical and mental health, and yeah.
Yeah, so I'm going to zoom out the lens and talk a little bit more about the study. So as Jagan mentioned, the objective of the study was to have people who are a particular set of participants and their exposome is common. What is the effect of a dietary intervention, which is short term, and these are particularly microbiome rich foods? The outcome that we're looking for, does this improve the patterns of microbes that is seen in their gut? Does it have any effect on their overall mood? Does it change the stool consistency? Does it also affect maybe their general overall behavior, which I think you're recording using mood, right? So in terms of the study timeline, there are people from across the world that have come here. So the natural question is that there are people who are from different parts of the world, so they don't have a baseline microbiome that you would expect that all of them would start with the same microbiome and then it shift. So at the beginning of the residency this week, the study started 2 weeks after these people arrived here, and they were given 2 weeks time to adjust to the local exposome, which will be the water in the ZuGrama, the kind of food that they eat here, what kind of environment they are exposed to. So the 2 weeks were baseline setting for these people, and then they all you would assume that even though their native microbiome will not change, the exposed microbiome might change in some way or the other. So at the 2 week interval, you will have people give a baseline sample that Jagan talked about. And then for the next 3 weeks, they will be eating the same type of food which is served to them. But out of that entire curated menu, they can choose and pick whichever types of foods that they want to eat. Some will be vegetarians and some are non-vegetarians. There will be people, so that's one kind of difference in these people's diets. Second classification is there are some people eating 1 meal, 2 meal, or 3 meals a day, and all of this information is recorded by them in an app. There will be people who are exercising and not exercising, and there are people who, as a part of another control group, we also have people who are living in the same setup, but they are not following the same exposome criteria. So we have a small group of people who are eating the foods that was prepared in their own homes. They go back to their own homes and their own environments, and then they are also part of this 3-week study where they essentially have no intervention. So you have an intervention versus no intervention group, and at the end of the study, all of these people will submit a post sample to assess the changes in the microbiome. Anything that I missed?
No, I think that pretty much is what it is, right?
Okay. So looks like once we take the baseline, a diet which is microbiome friendly, which could possibly see some improvements after 3 weeks. I have a new term which I came up with. I couldn't help but smile when I thought about it. Microbiomically gate kept. Okay. Right. So I'm going to put you both on the spot here a little bit, since we are into the study, right? Um, oh, those two. Um, okay. So now we will get into the details of what happens once you have the data, how it's processed, and so on. Both the scientific aspect, also just from an information perspective. If you zoom out a little bit, so when you guys were speaking, right? Our scientific curiosity is such that we want to see something there, right? How would you guys ensure that you're a part of a company who's interested in studying these things? Would you make the raw data available for other people to come back and analyze this if they wanted to? What sort of setups do you have to not have your own biases go into the study?
It's a unique position to be a private company,
Yeah,
right?
just to clarify, I mean, throughout history, we have seen where certain sponsors of the study, like skew the results, right, because of their maybe known unknown biases. I'm not suggesting that any of you have it, so that's why I thought I'm going to put you on the spot and ask you, how would you ensure that there are no biases creeping in, so you're not looking at things and picking out cherries that might help your company or product in some way?
Yeah, I feel that's a very important question, and I do understand that it's a private company, and usually we stand on the brim of all the things that you have just spoken about, right? Um, Most of the times, I like to think. I mean, I have mechanisms in my brain which I use to kind of address these important things that might come in my way, right? And that mechanism is that, do I think like a founder or do I think like a scientist, right? If I'm thinking like a scientist, I try to avoid as much biases as possible. If I try to think like a founder, I try to see how can I monetize something, right? When I decided to do this experiment in ZuGrama, it came out as a purely scientific curiosity, right? See, I don't do personalized cooking for someone, right? In my company. For me, it was just simple. I have been suggesting people that this is good microbiome friendly food. Please have it. Do I know enough about it? That's the curiosity. That's what I'm trying to prove here. And I feel we definitely want to publish this study when it gets ready, of course. And with the study, we have to make data publicly available. And we want people to test out their own technologies and pipelines on the same data. Because just like Jagan, I think in a very initial part of this conversation, he mentioned about something called reproducibility robustness, right? I mean, I could do all the things in my company, keep it hidden, and go out and make all that claims, right? But is that really helping science? And if that's not really helping scientific community, no matter how hard I try, I'll only be able to contribute 1% in the whole pool of things that are happening. So we do believe that there has to be a middle grounds to it. Yes, definitely there are some IP-able things which we keep in-house. There are some important data pieces which we keep in-house until we find something out of it. But once we find something out of it, once our motive is completed with it, the data is for the scientific community to find more things about it, right? And I feel all the things that I have learned in the past week out at ZuGrama about the DAOs and the decentralized systems and the ways to keep your data safe while it is open source, right? I think I would like to understand and use those mechanisms day in and day out so that I make sure that the data stays protected at the same time it is utilized for the good of the world. So that's my stand on it currently, to be very honest. About the inherent bias, I feel that if I have the onus of publication on ourselves, then we know that smart people will catch biases in this study. So we'll do our best. We are not the best scientists in the world, but we'll do our best to avoid the biases. And if someone else finds it, we are ready to address that.
Yeah, thanks. So we'll be able to publish raw data with some way, and it'll be open access for people to go read about it. Okay, this is probably a bit of a tangential question, but since we are here, maybe we can take it. In case you did, publish this data, what are the risks associated with it? Especially, you can identify a specific population with this set of, microbiome data. Can that be, biologically weaponized against a certain population?
Oh, about this data set, the unique thing is that there are people from all across the world, and once it is anonymized, it cannot be generalized for the Indian population. So with specifically this data, I don't think that risk exists. But towards your question of one country versus another country and how can you utilize somebody's biological data to weaponize it, I think we know very little about the microbiome itself at the moment. So unless we have more and more understanding of the microbiome and what kind of functions they perform and how can they be so weaponized, at the moment we are far away from it, but I'm not going to deny that the possibility that you present that in the future that there might be a possibility that bioweapons could be created out of it. You need far more complex biological data to do that, because you don't only need microbiome in isolation, you need the genome of the person, the entire makeup of the person, the kind of immune system they have, the kind of resilience factors they have, and all of this information from like a single person to be able to make those interconnected reasoning out of it, right? So that's the complexity that we're dealing with at the moment. So we can say that for now we're on the safe side, that potentially in the near future this is not going to happen.
Well, I'm not much worried about weaponization, but I've often seen that microbiome data is taken out of context to create products like probiotics and prebiotics, which they claim to be science backed but are never really science backed because they do not understand the intricacies of the data that has been used to produce that. I feel the current and more real problem is that unscientific product under the claims of scientific product, just because they have used some data, analyzed something, and trying to create a product out of it. And I think that the problems with the analysis and why data is often misinterpreted is something Jagan could answer better than me for sure.
I agree with that. Like, instead of weaponizing, I think people could put out very wrong products with respect to just saying, we've seen in the past that there have been products where people have just used, let's say, Western population microbiome data. And at those times, the methods to study the microbiome, and Jagan can touch up more upon that, were not very advanced to fully understand those results. And you cannot pick up Western population data, maybe one strain that was beneficial, and then sell it as a capsule to some another population, right? Or even in isolation, you cannot just do that. You need to first understand the intricacies, mechanisms, and the complexity of it before we can do that. So the non-scientific products is something that I would definitely agree with.
Interesting.
So coming to data, so these data are coming primarily from sequencing technologies. So there is a lot of different technologies, and you can generate a lot of different data from these technologies. The problem is people don't understand or don't really question what is the right data, right? So when I say right data, there is no such thing called this is right data and this is wrong data. It's more dependent on the objective of what you're planning to look. So depending on that, you will have to take a choice. So previously, when it comes to microbiome, right? So there was this method called 16S. So when I say 16S, it's an hypervariable region in a bacteria where you can uniquely identify what this bacteria is. So in this technology, you can just identify a bacteria. So it's just going to give you an overview of the population. So just bacteria alone, if you want to study archaea, it's called 18S, fungi 18S. So with this technology, the only thing that you can study is a population, not beyond it, right? You know there is, you could say bacteria is this is the composition and this is the abundance of certain species genus. You don't get to go till species. It's usually restricted till genus. That's the domain. But whereas currently, I think there are technologies, it's called whole genome shotgun technologies, right? So this technologies give you more range. When I say you sequence you sequence all the DNA that's present in an environment, and you don't just sequence bacteria, but you sequence bacteria, fungi, and virus. So in this technology, you just don't study the presence and absence of them in the environment. It's more of their pathways, right? What they do, functionally what they do. But also when it comes to these kind of technologies, right? Since you get more wide range of space to study in a data, you'll see a lot of noise. When I say a lot of noise, sometimes there is no understanding towards how much should I sequence, which is enough for my study, right? Sometimes you might not find certain signature in a study because you haven't sequenced the data enough. It's not that there is no such signature present in the data. It's just that you haven't really understood what technology and what kind of data you need. So most of the studies are biased because they don't answer this. They don't ask this primary question of, okay, what kind of data do I need, to answer such questions. Yeah.
Right, and I think just retouching upon that. The problem with creating bad products from good science, right? There's a very big group in India. Uh, I don't know if I should name it. I shouldn't. I feel I'll be targeted for sure. But there's a very big group in India uh, whose leader just passed away recently, and I'm very sad about that. But that group's product has come out of a study which says that it is a healthy prebiotic, and that is currently a standard used prebiotic in the whole Indian industry, which might not be a very good study to back a good product on.
Thanks.
Okay.
Thank you, Darshit. Thank you very much.
Okay, there's a bunch of questions I have, so I'm going to see which is the best way to take them all. But before going to that, you mentioned we have a control group already in the study, right? When you guys were trying to design the study, did you think of having a placebo thing inside the study? If yes, or if no, why not?
No, why? Because we didn't know what kind of people are going to be here, right? So when we were planning, we heard there is going to be another set of participants who want to be part of the study, but they don't want to, follow they can't follow the protocol. We felt that would be a good add-on. Like for example, let's say if I could find certain signature with respect to diet and microbiome, right? It will give me an additional validation if that was not found in that population, or if that changes was not found in that population. So I think it was incorporated into the study on later stages.
Also, I feel if we are talking about dietary interventions, it's more about having a negative control in front of the current dietary system that we are proposing for the study. Because placebo would mean that I'm giving them hollow food, which is not really a good practice when we are doing something like a community or citizen science.
Got it.
I would call that a control group, not a negative control group, because you're not giving them bad food to eat. They're just eating their own normal food. So I would probably call that control. But if they're not informed of, let's say, what is a good and bad food, could that still be a placebo effect, that I'm in the study and I'm doing something, maybe I should follow some healthy practices? Is that something that was communicated to this control group, or there is no change that they were told to do?
I think at least for this study, we have not particularly communicated to the control group about what exactly is a microbiome friendly and unfriendly food. So that might be it. I don't know. I just don't have enough knowledge at this point about how do I carry out a placebo-controlled study in microbiome dietary interventions. I think it's lack of knowledge, so speaking anything would be out of.
Yeah, because if you give them a certain type of food that's not necessarily microbiome friendly, but might cause like some other type of harm, then it affects your study as well. So it's better to let them continue with their own lifestyles.
Okay, so looks like you spoke a bit about sequencing, and looks like we'll have to collect a lot of metadata and see if there is coming up. Right. So you spoke about two different types of sequencing. My first question is, which one will you be using for the study? And the second one is, since we have a lot of data here, usually it just looks like to me from, I don't understand the science too much. Looks like to me there is probably a correlation that you can study, but not a causation towards what is exactly causing your, say, healthy outcomes, if there are any. So like, are you looking into something that might give you causation as well? If so, how are you going about it?
So to answer your first question, I think we'll be using whole genome metagenome sequencing. So we'll be
Is it the same as the shotgun you
shotgun,
mentioned?
yes, we call them shotgun too. And to give you more about the date. Yeah. So coming, so primarily we'll be looking for correlations. We'll be because we know what kind of we are collecting a lot of metadata. We would like to see what kind of community changes that we see through this metadata. So primarily is to just to check if there is any changes, right? And how rapid the changes are happening. Like for example, when people started the diet, how long did it take for the community to change? Then later we will be looking into, is it causing because of a certain diet, or is it happening, is it happening for certain people? Like, because the main cohort can have a lot of sub cohorts, because let's say it can't be just a diet, it could be other factors too, and we are capturing that metadata as well. We would like, so it's going to be primarily, it will be more of a correlation study, so that we can better understand where is the change, where is the change happening and why is it happening. The question is to understand that. So
Just to add on to that, I think the correlations can come between all the factors with all of the other factors. However, since you have a control group which is not taking this diet, and you have baselines for all of the people, if you do see a change with the diet in the certain number of people which is significant compared to the control group, you might have a causation with the diet as well. This is theoretical, however, and microbiomes are much more complex. So you may not even capture those kind of changes. So you may not be able to say that.
And I think microbiome studies are more so correlational in nature. All I'd be able to say is that at the end of this study, I would be able to say that this correlation is much more stronger than the ones we see in the other dietary intervention studies of microbiome. Causation studies in human microbiome are very, very difficult to carry out. It's because you'll have to go an ultra level of control. I remember a group in Germany who does causation effects of dietary polyphenols on the microbiome. What they do is they take a set of mice and they feed them strawberry day in and day out and nothing else for months at a stretch. For example, if I'll just replace all the buffet items today with just strawberries, right? How pissed people are gonna get on me and then you, Yash. So yeah, in fact, I would say that I think there's a fundamental science problem that causation studies in human microbiome are very difficult to carry out, right? And there are very serious interventions for that. So doing that is any which way is difficult. But I think stronger the correlation gets, the better it is for us currently for the overall field as well.
one main reason why we can't pinpoint a causation is because of a lot of variables. Maybe there are more variables that we are not, looking into it. But like he said, we should. The more strength we have towards correlation, I think we can move forward and see if that could be a causation. But still, there could be a lot of variables which we don't understand from where it is arising. Even though we call it as a control group, there will be certain variables that we are not actually, looking into it. So that's one of the main. And also you need, the more variable we take, the more sample size we need to find a statistically significant, even for a correlation. So I think causation is kind of challenging in microbiome studies.
Okay. All right. The reason I asked that is because I think we touched upon it briefly by saying something like good science can lead to bad products, which I think was studied using correlation between some data points,
Yeah.
right? So correct me if I'm wrong, but the current SOTA is you have engineered like probiotics, for example, to maybe treat like a gut disorder, right? I'm completely microbiome build at this point, right? I'm like super curious. Sounds like this biome is an entirely new world, which are like somebody used the word the other day as our puppet masters trying to, control
Yeah,
everything that
we are
we do.
not alone.
Yeah, we're not. Right. So first, maybe you guys could quickly give a download to all the listeners about like the current state of the art on what sort of gut related disorders can be treated using this, and then I have a follow up question to that, but I'll hold that for now and let you guys answer.
But before we go into that, I have a very curious question for Darshit. If I flush out all of the gut bacteria from, let's say, a mouse, will they live?
Inert microorganisms are available for microbiome studies, so they do manage to live, but we don't know how long for, right? So that's the biggest question.
Nobody's done that study in mice. I mean,
If
I'm not suggesting to do it on humans, but
yeah,
mice.
I think if they have, I am at least unaware of it right now.
And what about humans? We've heard about humans who've had like intestinal disorders, and a lot of humans where large portions of intestines have been cut out, and their stomachs have been directly connected to large intestines, for example. What about how does the gut adapt to those kind of individuals?
I think microbes are there for sure. If even if the small intestine is removed, right, the whole population of bacteria that might be living in small intestine might go away with that because those bacteria are, habituated to live or adapted to live in small intestine. But if there's any part of intestine that still exists in your body, that means it has microbes. It cannot sustain without that. And more importantly, because our gut has lining, or our intestine has the lining, right? The mucus layer and the whole immune layer. That's where most of the microbes reside as well. And if the microbe layer comes off, the whole gut kind of falls apart in a way. So it is absolutely necessary for structural integrity for microbes to be present in the intestine. If they are not, then there is a high level of I mean, the structural integrity of the gut would break down.
Yeah, and last question, because the question he asked is about the gut-related disorders. So previously you mentioned that breast milk has microbes that will help the child metabolize breast milk better. Is that
It doesn't
correct?
have the microbes.
Okay.
The vaginal
Ah,
microbiome
okay.
harbors bacteria which goes into the child, which will allow it to break down the
Oh, okay.
breast milk
So
character.
That is the
in that sense,
microbiomically gatekeeping.
um, so does that mean that CZ babies are not microbiomically gatekept, which means that cesarean babies will not be able to metabolize breast milk?
They will have difficulty because what happens is cesarean babies gets exposed to the hospital environment more than the vaginal microbiome. So the bacteria that's entering the infant who is coming from a cesarean delivery would have bacteria that are coming from the doctor's coat, or the tables around them, or the surgical instrument. So
Borderline scary.
Yeah, I wonder if this is in some way, like, for example, related to lactose intolerance developing
Yeah,
in some
that
people.
was my next question.
could be. There's a high possibility that could be. And in
Next
fact,
steady idea.
yeah, that is one
No, I do
thing.
have anecdotal evidence that people who are born from natural deliveries are also lactose intolerant, but that could be some other factor completely. But yeah, go
Yeah,
ahead.
actually, lactose intolerant is a genetic trait. It's not a microbial trait, right? But what there is one hypothesis I have. I have not tested it with data. Is that if you are genetically lactose intolerant, you will not be able to harbor lactobacillus, which is why you are having all the problems that comes with milk products. This is my assumption, and at least that's what I've seen as a pattern in the data that we have analyzed so far. But we'll have to test it out by doing genetic sequencing for most of them.
I forgot the question, so I'll ask whenever I remember.
Okay, so looks like we were getting to this
Disorders.
the sota of what can be done with, say, manipulating your microbiome to treat different kinds of disorders, or
I
whatever we
remembered
can
my
help with?
question. Sorry, this is related. That's why. Is there any exchange of microbiome from the placenta between the mother and the fetus?
Yes, there is. There have been some evidence about that, but the field has not reached a consensus on if that actually happens or not. Disorders are multiple. I think most of the gut disorders that we know of has a microbial influence to it, right? We know the celiac, we know the Crohn's disease, we know IBS, IBD, where mechanisms are unclear, but they are microbiome mediated. We have C. difficile infection, one of the most serious ones, I feel. We have ulcer formation, we have colorectal cancer. These are all gut related disorders which are directly, I would say, diseases as well, which are directly correlated to the microbes that live inside your gut. More so, Alzheimer's, cardiac issues, chronic kidney diseases, liver disorders. Again, there is a gut microbiome influence to that.
Is that um, is that an outcome or a cause?
I think there is correlational data so far. The directionality of it is unknown. If it is bidirectional, that let's say liver starts dysfunctioning, so the microbes change, or if the microbes have changed, which is why the liver has started dysfunctioning.
Yeah, so for example, if I take a step back, and we know that with age, the entire gut microbiome changes, and we don't know whether it is the microbiome change affecting aging outcomes, or it is the aging in the body which is affecting the microbiome. Similarly, in multiple disorders, what is fair to assume at this moment is that changes in microbial patterns can be used as a diagnostic tool for assessing these conditions, right? And also a fair amount of rebalance, getting the microbial conditions to an optimum state. Can that lead to reversal of some of these conditions?
The reason that we don't have much understanding about the disease microbiome relationships is the technological difference that Jagan was talking about, right? Because most of the studies so far have been done on 16S. Now, what happens is I'm thinking of an analogy to tell you, right? If I say that a group of engineers are present, that is why they can do this, it's much less, right? But if I can tell you what exact skills these guys have, right, then I can short predict that they will be able to do this job or not. And which is why the whole shotgun metagenomics come into the affair, right? Now, for the longest time when 16S was used to study, we thought that gram-negative bacteria, if they are present highly, then people have cardiac issues. But how? No one knows, right? Then we started getting into the genetic potential of it, the transcriptomic potential of it, which means what they are producing. Then we realized that, oh, there is a TMAO production and there is LPS production, which is done by bacteria, which enters the blood lining through the capillaries present in your intestine or through the breaks in the intestinal integrity. They enter the bloodstream, they reach the cardiac site, and then they cause an issue. Now, when technology improves, there is more scope of going down to these kind of understanding. Another example is we have a very constant member in our gut called Bacteroides fragilis. It's kind of a bacteria. Now, fragilis is known to produce a certain kind of toxin which causes double-stranded DNA breaks, which is the DNA present in the human cells. It breaks it down. And this genomic instability or breaking down of DNA is quite, I mean, not quite. It's a hallmark of colorectal cancer. It's a hallmark of aging. So now that the technology is improving, we are getting down to so much more understanding of what these microbes are doing to cause a certain situation. So now we are getting much more knowledge about the causation, or we are getting much closer to a stronger correlation of why this is happening. Similarly with the liver, right? So liver produces bile acids which enter your gut, right? And these microbes are known to transform these bile acids again so that the liver can reuse it. Now, these are some of the mechanisms which we never knew until the 2017, 18, 19. But now with all these papers, with this new technologies come, we are able to get to know that.
Uh, can I add another point towards microbiome and disease association? Like you said, initially it was just based on bacteria's and disease association. So that alone is not going to give us any strong correlation. Like we will still have a lot of questions, which direction. And whereas nowadays when people do whole genome shotgun, right? So it's not just the bacteria. You would get to know the pathways, right? What kind of pathways they have. And also coming to disease, it's quite complex because microbiome changes a lot. And second is each population, like if you see all over the world, the composition of gut microbiome is entirely different. And when it comes to each diseases, right, you should know what kind of like what kind of metadata you need to look into. You cannot have one a similar type of metadata and try to correlate all the disease and microbiome as it's not going to show a proper signature. So usually what people do is they take the studies are based on, like, you take a you take a disease population, you take a healthy population, and you see what kind of different associations are there, but not really they have an inclusion criteria or collect a proper metadata specific to those disease. So it's like you can't have a standard same metadata for all disease, because each disease are different. And obviously that is the reason we cannot actually see what is the causation or what is the direction for the changes of microbiome.
Right. Cool. Looks like we can manipulate microbiome to, like help with some diseases. But this is an out there kind of question. Okay. Can we think more differently and ask the question, can we manipulate microbiome to help with foods that is not usually consumable by humans? Like, can we change our microbiome or anything in the gut that might help convert things like cellulose into something that can be energy yielding, or even plastic for that matter? So this would give an entirely different option for humans to consume energy, which, if it works out, could possibly be a very different angle of attack for a solution set in food security as well.
I would definitely bet on it. From the Japanese study we discussed before, there is short evidence that genes can be transferred from an algal family to a microbiome to extract energy out of an algal plant. So the possibilities are endless. And if we could actually recreate that technology in an artificial way, artificial as in if we can actually leverage that technology to change your gut microbiome to get energy out of other sources, I think that'd be excellent. But I think the bigger challenge is not changing the microbial potential to convert something. It's about the challenge is more of, are humans able to consume that through their cells? Because microbes usually make molecules, right? But the real dependencies are human cells able to use that molecules. I think which is something that Suramya would be able to answer better as a cell biologist.
Yeah, I mean, if you're imagining this scenario where we can engineer gut bacteria and in the interest of not being microbially gate kept, I would assume that once you know the complexities of the microbiome and the potential of each of these species to break down one particular product into another, you can then harness different type of colonies that can coexist in these microbes, where one has the potential of breaking down, let's say, cellulose, plastic into another compound, and the other compound being utilized by the second species, being converted into a product. And this way we can reach a path where ultimately you have the final product that can then be utilized by humans to be able to be used as an energy source. But this is a very to get there, you need to understand the complex mechanisms of interactions between all of these microbes, and also understand that the conditions for all of this to go right are going to be very, very complex. So how to engineer that efficiently is something that we can think of. In theory, you can say that this is possible. Well, And not only about foods, what I was saying is that you can also engineer bacteria to have more resilience against certain type of conditions. For example, you could engineer them to produce a molecule or a metabolite that will give you heat resistance, cold shock protection. You could do it for exposure to radiation. You can give it to you can produce any metabolite that can go into the human blood, as long as that metabolite is safe and it is not causing harm to the human cells in any other way, causing toxicity in the body. That's possible.
While
In
she was
theory.
talking, while she was talking, an insane theory came up to my mind. If there's a microbiome scientist listening to this, please try this out. Okay. We know that FMT works. I'll tell you FMT in short. It's fecal microbiota transplant, which means pupils or capsules is two common words that are being used, which means that I'm taking poop from one person, converting it to a capsule and giving it to other one. This usually happens when, let's say, someone has a condition like Clostridium difficile infection, which they're not able to remove. So then a healthy person donates poop, which is then converted to capsule or pupil, or then given to the person with the infection, and that has shown insane level of remission, which has never been observed in microbiome sciences before. Now, the bigger problem in FMT is the engraftment issue. What I mean by that is, when I introduce a new community of bacteria, are they able to live in my gut or stay in my gut, or they just pass out, right?
Colonize.
Are they able to colonize or engraft into my gut? If I can make engraftment possible for all microbial presence in the world, then I definitely know that there are certain marine microbes which are able to digest plastic. I know that there are cattle microbiome which are able to convert cellulose to energy. And what if I can make a collection of multiple of these microorganisms, bring them down to a single community, and then find the right species doing the right thing and make a collective community? That means you might have to eat cow shit, if that's what I meant. But yeah, if somehow theoretically we are able to get all these microbes in a certain chain of events in a certain community, and then make sure that that community can go inside the gut and engraft, we'll be solving
Is,
issues that are
isn't it easier to take genes from them and then engineer them into the bacteria that can already reside in your gut?
for sure, but usually the mechanisms that are there, for example, a marine microbiome community degrading plastic to something else, is also a community feature.
Uh-huh. Okay.
Right? So it's not a single gene that is going to contribute to that. Just like you said, right? What if one microbe breaks down something, produces
Yeah,
something?
so the combination of colonies that you put in, can you use the host microbes that can already colonize the gut bacteria and then engineer it in a way where this particular community is able to sit in your gut bacteria, but the genes that it carries are from a mixture of all of these different bacterias. I know it's incredibly challenging, but you will cut out at least one problem where you have to solve the engraftment issue as well.
No, I feel so. Yeah, that is 100% possible. But let's just to be slightly critical of that idea. What if the engraftment of the species that are currently known to harbor human microbiome are there because of the current genetic makeup they have? And if you're going to change that genetic makeup, will they still be able to engraft or colonize in your gut?
I think that's a testable theory.
Yeah, of
I
course.
mean, we can easily test that in mouse models and use some native mice gut microbiome and then just change their genetic makeup, see if they're still able to colonize or not. Don't cut out like essential genes that they need to live in the gut, but take out another genetics, or you could introduce another copy. While we're saying all of these things, it's incredibly hard to do cloning and genetic engineering in the lab. So all of this is in a very idealistic world where we're just testing theories out.
I mean, everything that we have spoken in the last 2 minutes are great theories, but all of them are poop shots.
Yeah, they're poop shots. So we've got microbially gate kept, we have capsules, and we have poop shots.
Poop shots.
Pupils.
Oh, also, by the way, the microbiome study that's happening in ZuGrama is called the PooGrama experiment.
Okay. Yeah, looks like we would need some of the things that you were mentioning recently. Looks like an engineering plus a research problem. And we seem to be working on an FRO idea, which could be leveraged to solve these kind of things. Um, cool. And do you see a future where I think we didn't cover this earlier? Feel free to give some context over that. There seems to be a gut-brain connection. And do you see a future where such pupils can be popped in, and I can change my mood instantly?
It's just that there's not much evidence and regulatory backup to it. But I feel in very small community level experiments or people being frustrated by what they had been feeling have gone on to try this experimentally, and they have seen some excellent benefits anecdotally. So I think that is something that can even be done today. I'd be really interested to look in how mental issues could be solved, or mental problems could be solved with something like this.
But to take a step back and to do that using a pupil, can we touch upon the advantages and disadvantages of a pupil versus taking the metabolites directly? Because these are the change in the mood is going to be via a microbial associated metabolite. So why do we need to go through the complex process of taking the pupil, having them colonize, make them, let them synthesize a metabolite, and then feel the effects? Why cannot we just take the metabolite? And what are the advantages and disadvantages of both?
Yeah, before you answer that, can you also maybe give a very quick explanation of what a metabolite is?
Basically, things that microbes produce when they consume a certain kind of substrate molecule, they metabolize it and then they produce a byproduct which is called a metabolite. Some commonly known metabolites from our gut microbiome are acetate butyrate propionate. All of them are immune modulatory metabolites. Some others are serotonin, dopamine, and some of the GABA inhibitors that we know of. Then we have indoles and lots of other metabolites that we know of as of today. Addressing Suramya's question, right? What I have come to realize in a very short span of from what I've read about microbiome is that when I'm looking at diagnostic approaches for microbiome, I go more reductionist in nature, right? But whenever I'm looking for an intervention, I go more community because we do not understand the community dynamics as much. We do not understand if it is that one metabolite or a collection of things that are happening, right? So until and unless we are very much, it's just like the human metabolism problem, right? We have this big chart of complex connections and we still say we hardly know 20% of the human metabolism. I feel that's the same problem with microbiome. We do not exactly understand how a certain metabolite is produced except for some basic pathways that we currently know of. There are definite advantages if we can reach a certain level of knowledge, then I would also go metabolite level intervention. But for example, some of the postbiotics, which are currently being sold in the market, also are metabolites like butyrate and other stuff. They have shown pretty much almost less than 10% delta into the trials that they have been tried in. So we know that that approach currently doesn't work. So if I have to just put a consensus to the current literature on microbiome interventions, the least is the metabolite, the middle is the pre-probiotic sector, just above that is dietary interventions, and the most delta that has been shown is the fecal microbiota transplant or pupils. But since we do not understand the mechanisms of FMT also as much, the adverse reactions have been many in the current clinical trials that have been known, and only certain conditions have been tried. So yeah, there are some issues associated with each one of them, but as of where the current field stands, FMT remains to be the best option in certain microbiome-driven gut disorders.
Mm-hmm.
Go ahead.
Yeah, I thought maybe let's switch gears a bit. And I think we spoke about the timelines of this study, right? It's short term, and it might have some health benefits. We are unable to quantify that as of now. Um, so how do you think that this would lead into maybe this study like could help us understand long term health benefits? And are there any results that you think, that could have like public health implication advice usually as it's given?
I feel, yeah, the study will give out data which will be substantial enough to understand if a certain kind of dietary habit would help a person or not. And I feel the value has already started coming because I've been meeting so many folks who are on to the study. It has hardly been 7 days, and most of them have started saying. So there's so many intangible benefits I've heard from people around. Most of them are saying that my addiction to something like a Coca-Cola has gone down completely in a matter of 7 days. Some of them have started saying that I'm feeling more energetic than I ever was without actually taking that caffeine dependency at all. Some of them are saying that they had a lot of issues with bloating and the bloody stools, and that has pretty much gone down in a week as well. So I feel intangibly there are so many benefits. So I'm sure that the data is going to show something. I'm just running off from ZuGrama because I don't want to hear this anymore and get any more biases inside my head before I start looking at the data.
So, since I am someone who's coordinating this experiment with my team in Bangalore, so there is another layer above the experiment which I realized. So I know people in ZuGrama before the experiment and during the experiment. So I've seen few of them come up to me and ask, "Can you tell me what kind of what food is good? Like which should I eat, which should I not eat? Tell me." How do I balance when you say I have to eat this, but how do I balance it? So I've not seen this happen previous week. So so what I mean to say is that like if I mean to take a session on gut microbiome, health and lifestyle and the session. Let's say after an hour the session completes. I share my knowledge, but there is nothing happening beyond it. But here, if you see, I'm creating a space. I'm just when I say space, I'm more like it's like I'm giving you experience of science, not sharing the knowledge, right? I'm creating a space where people can come, right? They can try certain lifestyle habits, be it dietary and etc. So they can see what works best for them. So this is not just so when I say what works best for them, so this is going to continue just not just after it's going to continue even after experiment, even after ZuGrama, right? So they're just they want to know more about gut microbiome. They want to try different things and see what works best for them, and they have a lot of questions which they want to ask. Okay, why can you tell me, like, this is the diet that I used to follow, but I'm trying to do this, but I do see these kind of changes. So I think that's more exciting than the output that we are going to look later. So yeah.
In terms, sorry, yeah.
Go ahead.
In terms of the public health benefits, I feel that if this, I have a strong feeling that this is going to give me some strong understanding of how a controlled environment and microbiome behaves with a certain level of intervention. Then I would like to extrapolate this to hospital environments, army camps, or ships that and navies that go out on a certain environment and stay out there for long times, right? So I think there's so many applications then, because we can prove that in a controlled environment things can change to better. And then I would like to go into more clinical settings, or like the expeditions that different areas have. So definitely there is a potential for public good. I don't know how it's going to help a human live a individual life in a certain urban or rural area, but I definitely see that wherever there are controlled environments in this world, studies like these could become a framework for them to carry out their own dietary interventions.
I was actually going to add something similar as well, that if you use setups like ZuGrama, other zoo villages across the world, to have these very controlled experiments in lifestyle, food, different kind of habits that people find. Normally, what happens in any research study, you don't put people in the same room or the same house and let them feed on the same food, because there's a lot of implications in terms of people don't want to leave their houses or come together. But these kind of places serve as a very good framework, because then what you could do is what Darshan mentioned, is that in expeditions, in army settings, in space missions, in Navy settings, what you could do is you could also come up with better food that goes to them with this, with the expeditions. So if you have to design what is the best optimum food to carry with you on the ship, you know what kind of environment you're going to be exposed to. So this is the kind of food that you should take, which is the most suitable to the kind of people who are there on that expedition. So I think so far, everybody who undertakes these missions, they keep missing home, and they keep saying that, okay, my body is not at its best. But this could enable something like that in the future.
Yeah, that's very exciting to me as well, as to what does this mean for science. Like, looks like what you described is possibly like people first science. I think some people have another term for it, call it decentralized science, right? How do you think science might change if we were to extrapolate this into a larger community, right? Looks like now a community can come together, figure out what their microbiome is, figure out what sorts of food is healthy for them. They're able to contribute data to a system, and it's helping advance science and so on. How would you think this would look like if we expanded it into a larger scale, where you have bigger sized samples? So this could become citizen science first approach towards things where they have a stake in the game that's being played.
Institutionalized science has actually taken this away from the community, because most of the disruptive science has come from individual first and community first initiatives, right? Be it we talk about pasteurization, be it we talk about polio vaccines, or the latest one, the COVID vaccines, right? What are these? These are citizen driven science projects. Yes, the development was done by someone else, but then the community came together, accepted it, took it, and understood that there is a benefit. So I feel, especially with things like dietary intervention, I don't know why are we still adhering so strictly to the institutionalized science standards, because these are fairly easier experiments. We know that they are not going to have extreme adverse effects for sure. So if the community and individuals come together and do something like this repeatedly, I feel science will move ahead much faster in lifestyle practices than it has been moving in the past few
Yeah, I
decades.
think with science moving faster, there is a possibility here for you to, what I've been trying to name it as, completely different parallel science economy, where you don't have to be dependent on such institutions that you mentioned, where people are coming in, they're participating, giving their data, figuring out the science and research, and just implementing that already. So you have a very quick feedback loop that you might not have had previously.
I'm going to touch a little bit upon that, because even though Darshit mentioned that there are certain kinds of foods that the kind of experiment that we're doing here is diets and interventions, which include short-term changing of diets, and we keep doing that all the time in our own homes, right? For a certain period of time, we like to eat a certain kind of food, and then we move on from it. So if citizens can come together like this and contribute to even developing the overall frameworks, then I think it could have a lot of benefit for questions like these, because a lot of times in traditional setups where we do very focused experiments, we don't tend to do the effect of, let's say, diets or very broad questions. The science is extremely focused over there, where even in mouse models or in cellular models, we will test the effect of a particular treatment, which will be a single molecule, and how that will affect the health outcomes. That's very fundamental in nature, and that's also extremely important, because even though at a community level that will affect public health, at an individual level or a very focused level, you still need to know the mechanisms behind all of these studies, right? So while traditional setups can focus on the fundamental mechanisms, identifying what each of these compounds or each of these metabolites do and how they affect health overall, they can be taken up and that translation could be very long into humans. At a more application level, this kind of a framework can be utilized to understand more everyday problems, because in experiments which are very focused, you cannot do them on human beings, because if you don't know the mechanisms, you don't know the outcome. So you can't ask any particular human to just consume on their own a particular metabolite and then see how it changes, because what if it is toxic and it's happening in your own homes where nobody's finding out what the outcome was? So that becomes a ethical challenge, and it also becomes dangerous for the person to take that kind of an experiment onto themselves. However, with food, you normally keep changing diet, so it's generally fine to do those kind of things.
Yeah, I'm sort of personally excited about, that's like one way to think about it, right? So you're trying to have a certain set of, solution set that we have accepted as, okay, this is what we have, and then trying to modify the microbiome in a way to make it healthy for humans. But the other way to think about it is how do we, if we can alter the microbiome to process things like cellulose, it might change the game for food security, right? Especially if in case we want to have people go into an area where our dependency on certain types of foods is not available, then we can actually change the human who is dependent on those foods, rather than trying to provide certain set of foods for people as well. I'm not sure how much time this would take and whether this is even possible, but sounds like an exciting future.
No, I feel that's definitely a very exciting area. I mean, I don't see much of a challenge with the microbial communities adapting, because just on a time scale, if I have to put, right, a bacteria divides or evolves more than 100, 200 times, right, in a very small time scale. So their adaptation to environments is pretty quick. Imagine it's growing in Chernobyl sites, right? Still, that's insane. Now, the problem is that they will survive. The problem, the solution is that they will survive, and let's say we are able to find them to inhabit our gut as well. I still see a bigger challenge with the host cells accepting most of the things that are happening, right? And I think I'll need Suramya's help to solve that bit of it while I solve the microbiome.
Yeah, we've also not touched upon the health of the host in this entire experiment, right? So while they are interdependent, and in some way one will affect the other, but does optimizing the gut microbial community and the kind of the bacteria or fungi or viruses that inhabit it, that does not necessarily mean that it will lead to all beneficial outcomes if the host is not suited to take it up. For example, with age, there are huge amounts of changes that happen in your gut lining. So if your gut lining is there is the barrier integrity is compromised in some way or the other, then that means more and more pathogenic bacteria have a chance of invading your bloodstream. Or if the barrier integrity is not intact, then that also means that whatever metabolites are being produced by these bacteria may not even be absorbed by the host. One classic example of that is that in a lot of individuals that have inflammatory conditions, where the immune system is hyperactive and there's some sort of a issue with their gut linings, you can keep replacing the bacteria in the gut with vitamin B12 producing bacteria. However, the vitamin B12 will not go into the bloodstream. So in that case, you need strategies like, okay, do I give sublingual vitamin B12 or not, because it's not going via the gut. So and so in those cases, I would assume that studying the host becomes important, and what kind of interventions can I do along with my capsules to make sure that the barrier integrity is then intact, it is healthier, and how do I enable the most efficient absorption of all the metabolites that are being produced in this bacteria as well. So I think along with the understanding of the microbes that are living in the gut, we also need to understand how the host body itself is changing, how those are more or less suitable to absorb some of these beneficial metabolites or not.
Yeah, that sounds super interesting. I'm like interested a bit in food security because of the other work that I do, and I was curious to know a bit about if this can't be done for humans, can we do it for other animals? Mainly because some of the animal food that animals eat today, humans can actually consume them if we, if we had a crisis or a catastrophe scenario. So can we are the challenges simpler in other animal systems where we can alter their microbiome to possibly eat something else, and humans could actually potentially take that in cases where we have food shortage.
I feel the day we achieve something like this in the first organism, from there to the human journey is not that difficult. So that's what I know. That's what I've known about any kind of science that we have done. Right? It's if we can do it in one animal to start with, it could be a rodent, it could be a mammal. I think we have pretty much won a certain level of game, and then we just need to keep capitalizing on how to translate.
I wouldn't be so optimistic on that because we're still, like for all the mice in the room, yay, we've cured cancer, but we've not been able to do that in humans. It purely depends on the complexity of the problem and the host interaction with those kind of species as well. So doing it in one animal's but your question was different. If you're able to achieve it in another animal, then yeah, the humans could just go ahead and consume them, which becomes a different question altogether. I'm not sure what kind of animal it's going to be, though.
Right. Okay. I think we touched a bit upon long-term health. What sort of effects do you think, say, if we are able to magically make some progress on research tomorrow, how does it help with studying aging generally for humans?
Microbiome research specifically? Okay. Yeah, so there's we've not listed out all of, Darshit has touched upon what all beneficial effects microbes have on the process of human health. But specifically we have very poor understanding of the same with the process of aging, and largely that's because we don't understand aging bodies themselves. So tomorrow, if magically we were to advance science in a way where we would know more about microbial species, we would also need to magically know more about the host organism. But assuming all of that is done, I think the scenarios that you were describing where engineering microbes or finding that optimum balance is achieved, you could possibly see immune modulation, where optimizing your gut bacteria would lead to better immune outcomes, because there is a huge amount of evidence now pointing towards that an imbalance in gut microbes, let's say in conditions with metabolic disorders, with obesity, are linked to an effect on the immune system. And when you change that, you have changes in the immune system itself. We could have a vast amount of effect on mental health benefits because we have the gut-brain axis, which is connected. There is also going to be a lot of effect on overall improvement in organ health, because when you have optimization of different kinds of metabolites in the blood, those would then improve the overall functions of the organs themselves. So I would assume that would look like a very good scenario overall. However, the challenges still remain understanding the host completely and understanding the microbes completely. And then once you have all of that information, both could co-evolve together or could be engineered to co-evolve together in a much more optimum way.
Highlight 4 experiments, 2 we have done, and two somewhere else it has happened. One was on a mice, on a group of mice, not a single mice, of course, done somewhere in England. Yeah. Sorry about the English. Yeah, but yeah, one was done in England, where what they did was they took poop of young mice, put it in old mice, and then poop of old mice and put it in young mice. So both ways. And when they took poop of young mice and put it in old mice, certain hallmarks of aging reversed. And when they took poop of old mice and put it into young mice, the young mice started behaving like an old mice. So there's short evidence that longevity, aging, and microbiome has certain kind of connection into it. Second study, was on Centenarians, right? So it was a meta-analysis where they took uh, lots of data from studies that have been done on people living 100 years and above, and then they saw that the elders who were comparatively healthier had different kind of microbiome profile and metabolic profile compared to the people who were unhealthy after 100 years old, particularly in two places where folks who were healthy and above 100 years old had a certain bacteria called Bacterium limosum in them, irrespective of the geography, by the way. And the second point was that their proteolytic potential, which means the ability to break down protein, was higher than saccharolytic potential, their ability to break down carbohydrates or sugar. So these are things we know from external studies that have been done outside. Two studies in house. One is our own mice study. We are still analyzing the data on that. So hopefully we'll get some answer from it and we'll publish that sooner than later. Second study, which actually got completed day before yesterday. Again, data something we'll publish, but it was more so to use some computational biology techniques to create a stronger theory. I'll not say proof, but to create a stronger theory about it. And where we definitely saw that we had a list of about 7, 8 bacteria, right? Some metabolites that they produce, which directly can target the innate proteins we know, or proteins or biological targets we know in host, which are strongly associated with aging. So I think these are four on top of my mind, and two since I have done, but I think lots of studies are there similarly. So there is that preliminary evidence which can take us to a strong hypothesis that microbiome and aging are intricately connected. Exactly through which mechanisms, I think we'll get to know in the next few years as more and more studies come out.
Yeah, it would be crazy if poop was the answer to our problems, right?
Betting on that.
But in the examples that you're mentioning, you're talking about mice to mice, humans to humans. Like, what about
No, don't
just
say it.
without me saying it? I think you all got it.
I would definitely bet on that as well. Like I said, I think one of the theories we were discussing, I told that I know that cows have the human microbiome which can digest cellulose.
What are some animals that we know lives really longer compared to humans? Which one? Turtles?
Only certain kind of turtles. I know certain kind of sharks. Greenland sharks live very
Yeah.
long, right? Yeah, Greenland sharks live very long. But I'm not sure marine microbiome is going to work on us. So we'll have to find some terrestrial animals that lives comparatively longer. I think bats are a great example for a size of.
The last time a human ate a bat.
Oh.
Well, that's what they made you think. Okay, all right. Yeah, we are going into the frontier, which is very exciting. I wonder if you've looked into any prehistoric um, data about this. Like, is there poop available from dinosaurs somewhere, which you can dig out and do some studies on?
I think there are some fossils for it, right? But I'm not sure it's viable enough to be consumed anymore. Yeah,
Okay.
but
You
what if
might
I?
find something that is preserved well,
Mummies.
hidden
I
in
think
somewhere.
permafrost microbiome is very interesting thing that people are studying.
Oh,
So
yeah, yeah, yeah.
yeah, so
Mhm.
permafrost microbiome is definitely something we should be
Can you
studying.
tell what that is?
So permafrost is ice that have been frozen near the poles for since like early Earth days. So I'm talking about millions of years, right? Since the whole planet started cooling down, and lots of people are saying that when that is, since because of the global warming, though that permafrost is melting, lots of new microbes are entering the polar water systems or oceans.
It reminds me of this series on Netflix, which is called Sweet Tooth,
Oh, yeah.
where this thing goes hunting for like this what they call universal blood, which is only available near the poles. Sounds something very sci-fi like that. Cool. I guess we have covered a few points, scratch some surfaces. I think maybe we should do another one to get into more things about the gut-brain axis, whether there is a relationship between intelligence and the kind of microbiome that we can host. Any closing remarks from all of you?
Let's do the dung pill experiment in the next ZuGrama.
Not be a part of it for sure.
I'm pretty sure this has to be cut out. ISKCON would be interested to sponsor something like that.
Actually, we shouldn't cut it out. We might just get funded by them.
No. Yeah. Closing remarks. I think there's a lot that we don't know. So that means for the scientists, more and more research studies, more data. We need a lot more information. But I'm sure with the advent of new computational powers and the AI technologies that are developing now, we'll soon be able to figure out some of these complexities and then advance towards a better future where we can harness the power of microbiome to optimize better humans, and how both of them can co-evolve. And can we find some answers to age-related diseases and also aging in a healthy manner in the microbiome? That's what I'm excited about.
Definitely, and I think we should do a special session on the whole data problem of the metagenomics, why there is so much issue with the current data that exists about microbiome, so that then Jagan becomes suddenly so articulate that all of us would be kept aside.
Yeah, I think I need to do a one-on-one podcast with Jagan to get him to articulate. Well, thank you so much for your time, gentlemen and lady. Really appreciate it.